Hello, Life Science. Hope you're doing well. Today we're going to continue Chapter 6, Role and Components of the Immune System with Documents 3 and 4. Document 3, the non-self. What is the non-self? It corresponds to the foreign elements that invade our body. The figures you see represent disease caused by the infectious agents like streptococci bacteria and coronavirus COVID-19, which both lead to pneumonia, which is characterized by serious lungs inflammation. Also, chickenpox is another disease represented in the second figure, which is due to varicella zoster virus. So the virus is also a non-self. Another disease represented in the third figure, which is measles, that is caused also by measles virus. So viruses and bacteria are famous examples of non-self, but are they the only ones? So what is the non-self by definition? The non-self is the foreign element that is recognized and rejected by the immune system. But actually, does the immune system recognize directly the non-self, like the bacteria, for example? Actually, the immune system recognizes parts of the non-self called antigens. So antigens are parts or molecules of the foreign elements. What are these antigens? What are they made up of? They are usually proteins and polysaccharides, and less frequently lipids, and usually these antigens are represented or displayed on the surface of the non-self. So, what are these antigens? These antigens can be cellular antigens, which means antigens displayed on the surface of cells. Example, the antigens that you find on the surface of red blood cell. For example, here, an individual who is A positive presents on his red blood cells antigen A and rhesus antigen. Why they are called antigens? Because they are considered by an individual with an incompatible blood group, like group B, as non-self. Also, these cellular antigens are found on bacteria. Soluble antigens, unlike cellular antigens, the soluble antigens are not located on the surface of cells, but they are soluble. Example, the toxins released by bacteria or present in the venom of snakes and bees. So here are toxins produced by bacteria. Also, the antigens could be a particle like the virus represented in this figure, COVID-19 virus, and pollen grains that contain the male gamete of flowers. Other examples of antigens like the vaccine, which is a harmless substance, and the vaccine is going to be discussed in Chapter 8. So, the modified self is also considered as non-self by the immune system. What is the modified self? Before explaining what is the modified self, let's remember together what is the immunological self. It's when a healthy normal cell displays on its surface HLA class 1 with a self-peptide or a self-protein. Of course, this complex HLA class 1 self-peptide, known as immunological self, is tolerated and accepted by the immune system. Now, what is the modified self? This cell, which is a self cell of your body, is, let's say, infected by a virus. So this virus, when it enters inside the cell. It produces viral peptide. The viral peptide is called a non-self peptide, will be bound to HLA class 1 and together will be displayed on the surface of the cell, and thus they form the modified self. Now we're done with document 3, the non-self. Let's start talking about the cells of the immune system. The immune system consists of several organs and cells. The cells collaborate with each other to ensure the body's defense against aggressions. What are the cells of the immune system? These cells are the white blood cells, also called leukocytes. Origin of leukocytes. Where do these leukocytes come from? Or where are they produced? They are produced in the bone marrow. Then, what is the bone marrow? The bone marrow is a spongy tissue in the middle of certain bones. Most blood cells are made in your bone marrow. This process is called hemopoiesis. It happens in children where in the long bones like the thigh bone. However, in adults, it is mostly happening in the spine, vertebra, in the hips, in the ribs, in the skull, and in the breast bone or sternum. Okay, then how are white blood cells produced? This is the bone, and this is the bone marrow that contains hematopoietic stem cells. These stem cells are pluripotent stem cells able to divide into different kinds of cells. So first you see that these hematopoietic stem cells, they divide into two types of cells, the myeloid progenitor cells and the lymphoid progenitor cells. So, the myeloid progenitor cells, what do they produce? They produce the first type of white blood cells that we call granulocytes that are formed of eosinophils, basophils, neutrophils. A second type, which is the monocyte. However, a third type of white blood cell called lymphocyte is produced by the lymphoid progenitor cell. And these lymphocytes are of two kinds, T cells and B cells. And the last type of white blood cells is called mast cells and is produced by the myeloid progenitor cells. Let's talk now about the morphology of the white blood cells. How do they look like? The basophil, neutrophil, and eosinophil that form the group of granulocytes, they all have a multi-lobed nucleus. You see clearly the nucleus that is not round, it is made up of many lobes, many parts. The monocyte, when found in the blood, it is rounded with a horseshoe-shaped nucleus. However, when the monocytes move in the tissues, they transform into macrophage. When do monocytes move in the tissues? This happens after infection, which means after contact with the antigen. The small lymphocyte is a bit different from the other white blood cells. It is rounded, smooth, because it doesn't have granules like the monocyte and like the eosinophils. It has a large nucleus and the reduced cytoplasm. What are the two types of lymphocytes? Lymphocyte B and lymphocyte T. And both lymphocytes carry on their surface specific receptors. Let's see what are these receptors. First, let's talk about lymphocyte B. The lymphocyte B carries on its surface membrane-bound receptors called BCR or B cells receptors. Membrane-bound means that they are bound to the cell membrane. These receptors are called also antibodies or immunoglobulins. After contact with the antigen, these lymphocyte B transform into plasma cells or plasmocyte. What's the role of plasmocyte? The plasmocyte secretes in the plasma antibodies, and these antibodies are thus soluble and not membrane bound. So, the antibodies, also called BCR, can be membrane bound. It's when LB did not meet the antigen yet. But these antibodies become soluble after contact with the antigen. What about lymphocyte T? The lymphocyte T are divided into two kinds. The first kind that we call T helper or T4. T helper, H helper, because this type of lymphocyte helps other immune cells, and this will be discussed in chapter 7. T4, because this lymphocyte T displays on its surface a receptor called CD4. In addition, of course, to the main receptor that is called TCR or T cell receptor. As you see, TCR is membrane bound. The second type of lymphocyte is TC, also called T8. Why TC? It refers to cytotoxic or killer, because TC kills infected cells. Why T8? Because this type of lymphocyte displays on its surface, in addition to TCR, another receptor called CD8. You see here also that TCR is always membrane bound. So the TCR is always membrane bound and can never be soluble like in the case of antibodies. Thank you for your attention. See you.